Today we'll be presenting to biotechnology and specifically forensics. Forensics is the use of gathering information to piece together past events, and DNA fingerprinting is the main idea or subtopic in biotechnology with forensics. 99% of humans share 99% of their DNA, and the 0.01% is used to identify, is the variant DNA that is used to identify people, and DNA fingerprinting is the way of use of identifying different people. And DNA fingerprinting is used in categories such as taxonomy, molecular archaeology, and paternity testing. However, there's some problems with this DNA fingerprinting, such as in the areas of ethical, social, and political ideas. However, they can be refuted and disputed, which we will show at the end of the PowerPoint. I'd like to bring over Michelle for DNA fingerprinting.
Forensic pathologists are able to use biotechnology to rule out suspects. This is because, like what Vivek said, 0.01% of DNA of individuals is different. This amounts to 3 million base pairs. The two processes in which forensic pathologists use to rule out suspects are PCR, which stands for polymerase chain reaction, and electrophoresis. Like I said before, the 0.01% of DNA amounts to 3 million base pairs, which is enough for scientists to use. The purpose of PCR is to create multiple copies of the polymorphic region. The polymorphic region is a section of DNA that scientists want to study. It is important because if DNA is contaminated, there are multiple copies to back up. The following are steps for PCR. To begin with, the extracted DNA, primers, DNA polymerase, and floating nucleotides are placed into a PCR tube. This PCR tube is then put into a DNA thermocycler. The DNA thermocycler rotates temperatures periodically. The three stages of PCR are denaturation with the temperature set at 95 degrees Celsius, annealing, which has the temperature set at 65 degrees Celsius, and extension with the temperature set at 72 degrees Celsius. These temperatures are varied based off of this length of the polymorphic region. The primers hold the DNA apart while DNA polymerase matches the floating nucleotides to the DNA evidence. The next step of identifying if a suspect can be exonerated or not is electrophoresis. The purpose of electrophoresis is to search for band patterns among the DNA evidence and separate DNA strands of different lengths. The steps of electrophoresis are the following. To begin with, DNA is placed into a tray along with agarose gel. Agarose gel has wells that allow negatively charged particles to move through. An electric current is placed on the tray. DNA strands move from the negatively charged side to the positively charged side because opposites attract. Strands of the same length clump together. The short clumps move faster and the longer clumps move slower. Therefore, over time, the shorter clumps are at a farther distance than the longer clumps. Scientists use a dye called ethidium bromide to dye the DNA evidence so it can be seen under the naked eye under an ultraviolet light. The following is a diagram of what electrophoresis occurs. Now I would like to hand it off to Namin.
Archaeologists use forensics to their day-to-day lives to identify and piece together past events of previous human life and other life. Biotech is highly prevalent in archaeology as well. Biotech was mainly first discovered when DNA was discovered in ancient bones, and the analyzation of DNA found in ancient bones was what really started the use of biotech. Similarly to how humans are DNA fingerprinted today, ancient bones and ancient people that archaeologists find are also DNA fingerprinted. The DNA that's found in the crushed bones of these people is very useful in identifying stuff about these people. And one of the main things that's useful about this DNA and analyzing the DNA that comes from archaeologists' finds is determination of sex. Prior to the discovery of DNA in skeletons, archaeologists relied on skeletal structure alone to determine what sex ancient humans that they found were. However, now that they can analyze the DNA that was found in these skeletons, they can look for what the Y chromosome, and they have a more accurate understanding of whether or not it was a male or female. However, DNA can only be retrieved in special circumstances. Although DNA lasts for about 6.8 million years, it only remains readable for about 1.5. So paleontologists and archaeologists have to find DNA that's within that time period. One main example of DNA, of really old DNA that helped in the field of archaeology, was the DNA found under Greenland. Greenland is covered in ice. However, they found 400,000-year-old DNA underneath Greenland from plants and insects, which suggests that Greenland actually once had greenery and wasn't always so icy. I'm going to pass it on to Sahitya for more on paternity and forensics.
Paternity tests are used to find relatives or ancestors or a potential father for a child. These tests look at non-coding DNA, which is DNA used that do not have codes for the cells, and they use this DNA because a human genome has 75% of non-coding DNA. It can also be used by STR, which stands for short tandem repeats, and you look at the repeats and then create a DNA profile. These profiles are used to see the matches for all the people. In a standard parental test, the people that are tested are the child, the alleged father, which is a biological father with 99.9% accuracy, and the mother. And I'll pass it on to Joe for taxonomy.
Alright, so forensic taxonomy is taxonomy in general is the classification of plants and animals and other organisms through phenotypic and genotypic evidence, which is DNA tests and physical traits. Forensic taxonomy is different from regular taxonomy because it strictly relies on genetic variation and differences between two organisms. An example of this is, just recently, the African elephant was subdivided into two species, the African bush elephant and African forest elephant. This is because after scientists observed their DNA sequences, it showed that they were very different instead of the same, almost identical as it would be if they were all the same species. So they had to subdivide the species. The DNA was examined because the physical traits between them were not clearly shown, but it was known that there was no gene flow in between the two populations. Several species have been broken down like this due to further tests done in the field of forensic taxonomy. This is because of slight differences between the two organisms' genetic makeup. There are a few problems with this, or controversies that people think about. There are no real ethical problems to taxonomy because it's just the observation of DNA strands to compare two different organisms, but there are also social and political problems with it. One social problem is that some people believe that it's not fair that the differentiating between two organisms relies solely in the hands of the scientists examining the DNA and other scientists that observe their results. This is because that they see that not every scientist observes the same two strands of DNA and classifies them as different species or the same species, because there is no set in stone rule about the way that the number of differences that create a different species. Because of this, there is no way for one scientist and another scientist to conclude the exact same thing with the identical results. This is possible because scientists may have the same classification strategies, but people find it a controversy politically and socially because they do not believe that there's not enough of the same beliefs to have all the species be classified exactly the same and separated into two groups. Now I'm going to go to Michelle for DNA fingerprinting.
If you're wondering, what if DNA is destroyed, and then you can do a DNA fingerprinting test? There is a solution to this. Scientists are able to look at the mitochondria to look for DNA. Regular DNA have things called short tandem repeats, like Sahitya mentioned. These are repeats and sequences in the DNA. Scientists are able to piece together mitochondrial DNA to form these strands of DNA that give the same information that DNA has. It is essential for DNA to be handled carefully, otherwise contamination occurs. But if it does occur, we do have a solution for that, which has to do with PCR and the multiple copies. DNA fingerprinting can be used as evidence in a courtroom as long as the people who are interpreting the information and the lab technicians are not biased. Now I will hand it off to Namin.
Some more political problems with DNA fingerprinting include people's belief that DNA fingerprinting infringes on their Fifth Amendment and their right to self-incriminate, or the protection of the right to self-incriminate. Some people believe that when their DNA is taken and presented as evidence in the court of law, it is infringing upon their Fifth Amendment right. However, we as a group disagree with that view. We agree with the judges and the courts, which is the state makers in the decision. We believe that DNA does not infringe on your right of self-incrimination, as DNA, as the courts rule, that DNA is not testimonial, meaning that it serves no testimonial purpose in the court of law. So DNA simply serves as evidence.
The following here is a social controversy. The FBI, which is the Federal Bureau of Investigation, holds a program called the IAFIS, which stands for the Integrated Automated Fingerprint Identification System. This program holds the fingerprints of criminals and civilians working under the government. This database is a controversy because people argue that this system is against their civil liberties, which is a value that is given up. On the contrary, this database is critical for the government to be able to fortify homeland security and fight against terrorism. Now I would like to hand it off to Namin.
A certain ethical problem that may arise from DNA fingerprinting is what we call cold hits. Cold hits are when there's a crime and DNA is the sole evidence that connects one person to a crime, and that's called a cold hit. And the controversy with that is it excludes any other evidence and puts DNA, which is the simple matching of loci, as a superior piece of evidence in the court of law. And this is considered hugely controversial. However, we still agree with the opposing side. We believe that DNA simply serves as evidence in the court of law and can help find guilty people and can help free innocent people.